Hello. Today we're going to be extracting chlorophyll and doing a thin layer chromatography. This is a more advanced form of chromatography than the paper chromatography you may have seen before. To do this we're going to use a special medium. Okay. Now this is a very small, but this is a thin layer chromatography plate. Okay, so instead of it being a paper, it's a layer of plastic which has had a very thin, it's a bit like paste or icing painted onto it. And it's a special substance that's very even throughout. Okay, so it means that when you run a sample up it, you get a very even and continuous density and movement throughout the plate. Now, this stuff is very fragile, okay, and because it's such a sensitive material, it's also very sensitive to fingerprints, grease and dirt, so you must make sure that you keep it exactly clean, and when you handle it, you only handle it by holding the edges, okay, so you can hold the edges top and bottom, okay, and be careful not to bend it. It's very easy to put too much pressure on and bend it. If you bend it, you'll create cracks in the medium, which will actually prevent the sample from running correctly. Now, what we're going to do first of all is we're going to measure up, okay, in this case 1 centimetre from the bottom. It is a very small plate, but it's okay for what we're doing today. Now we're going to do this using a graphite pencil. You can't use ink or anything on these plates. Now you must do this very, very lightly. Okay, and you're literally going to do a very faint, and I do mean faint and light pencil line across the surface. If you try and use too much pressure or too much force, you will literally rip a line right through the media down to the plastic all the way across. So you're literally doing the gentlest, faintest pencil line across the top that you can. Okay, now what we're going to do next is we're going to extract our chlorophyll. And we're extracting chlorophyll today from the chloroplasts within inside this spinach. Okay, and then we're hoping that we will see the different coloured types of chlorophyll extracted out onto our chromatography plate. So we're going to put our spinach into a small pestle and mortar, and we're going to add a small quantity of sand. Now we're just using the sand as an abrasive, okay, to help us break down those cell walls. If we don't manage to break down the cell walls, we will not manage to get the chlorophyll out, and therefore we will not get the sample onto our chromatogram, and it will not run at all very well. So you really have to work with the sand and with the pestle and mortar to really break this down into quite a green pulp, and you'll start to see that there will be small amounts of green liquid come out in the bottom, but it's a very small amount. So when we've done that, we would add a small amount of an extraction solvent. So the extraction solvent we're going to use today is propanone, which is also known as acetone. Okay, it's a highly flammable chemical. So obviously there's no flames or anything else around, and we have our safety glasses on. So when we've managed to mush up our spinach, okay, we're going to add a very small amount, and you're literally talking 1 or 2 mils of propanone. Okay, if you don't want to put too much in there, because then that will make for a very dilute solution. We're then just going to work it around a bit so that we can make sure that we get a reasonable amount of chlorophyll into that solvent, and you'll be left with a very small amount of a green liquid in the bottom of our pestle and mortar, looking something a bit like that. Okay. We're then going to use a capillary tube, very carefully handling these because they are very thin glass tubes, to extract the chlorophyll from our sample. we're going to hold the capillary tube in the middle with our finger up. We're going to put the tube down into our sample, okay, and then we're going to put our finger over the top of it and pull it out, and that will stop the liquid running back down it. And as you can see, we have a green solution has been extracted. Now in this case, we're just going to do one spot. So we're going to take our TLC plate, we've got our 1 centimetre line across the bottom, and in the middle of that 1 centimetre line, we're going to just spot on top some of our chlorophyll sample. Now, we need this sample to be concentrated, okay? The more concentrated we make it, the clearer the bands will be when they've separated out. If we don't make it concentrated enough, we may not see all the bands. And that's why we're using propanone as an extraction solvent, because propanone will evaporate and dry very quickly on the surface of our TLC plate. So we put our sample on, we're going to leave it, and it will dry within 30 seconds to a minute or so as the propanone evaporates off, and then we're going to repeat the process. Now you're going to have to do this about 10 times, okay? The more concentrated you can make that spot, the more likely that you will see all the bands and that they will be clear. Okay? And when we've done that about 10 times, we're going to leave it to dry so that we know that the sample is completely dry. We're then going to get a tank. Okay? Now, we're going to put our extraction solvent in there. Sorry, our chromatography solvent. Now our chromatography solvent is a solvent which is going to run up the plate and is going to carry our chlorophyll material through the plate. Okay, in this case it's petroleum ether and propanone on 7:1, so that's 7 to 3. 7 parts ether to 3 parts acetone. Now we're going to put this very carefully into the bottom of our tank. Now, it's important that we do not get to the 1 centimetre line on our TLC plate. So you're only actually looking at putting about 5mm depth of solvent into the bottom of the tank. If the level of the solvent reaches the line and the sample, instead of the sample running up the plate, it will run out into the solvent, and then you will not get any results from your chromatogram. When we've done that, we're going to very carefully, holding it by the top, drop it down into our solvent to run. Right, while it's running, we're going to put the lid back on. Okay? The reason we put the lid back on is that we want the solvent to run up the plate. Okay? If it's open to the air, as the solvent's travelling up the plate, it will evaporate off and dry, and it will not make its way the whole length of the plate. Now I've got one that we did earlier, which is a little bit faded and battered, but we'll show you it anyway. So, as you can see on this plate possibly, we have 2 spots originally and 2 separate lines. We have a very faint pencil line across the bottom, and then at the top you can just see that we stopped it just before it got to the top. Okay? Now this top line is the solvent front. So when the solvent has travelled up the plate and we take it out, okay, and we have to catch it before it gets off the top of the plate, we will then draw a line where we see that the liquid has got to. Okay, this is called the solvent front. And by measuring between the 2 lines and between the line and the sample, you're able to work out what's called the RF value and then work out what the chemical is. Okay, so this one is actually running very quickly. Okay. These would normally take anywhere from 10 minutes to 40 minutes depending on the size of the plate to run. So we're not going to leave it to run, okay? But that's how you would look at it afterwards. Thank you.